Hello students, today let's discuss about molecular basis of mutation. In introduction, mutation is the change in the structure of a gene which results in formation of variants that may be transmitted to subsequent generations. It may be due to change in a single base unit in a DNA, may be due to deletion, insertion, or the rearrangement of a large section of genes or chromosomes. Since the change takes place in a gene that is present in a chromosome, it is often referred to as a point mutation. There are various ways that point mutation can occur. Most of the point mutations result in loss of function of a gene, as it is easier to break down a machinery by altering its function than enhancing its function by making minute changes. As a result, mutations that produces gain of function are much rarer. Let us understand the changes in the DNA leading to point mutation. At the DNA level, mutation can arise due to base substitution, addition or deletion. In base substitution, one of the bases is replaced by another base, which again may be classified as transition or transversion. In transition, there is a replacement of purine by another purine base, whereas in transversion, there is a replacement of purines by pyrimidine bases or vice versa. Addition or deletion of bases is another type of point mutation that we will be discussing today. In order to understand mutation, let us look at the consequences of mutation. Mutation alters the codon of a gene and directly affects the polypeptide that is the product of the gene. In single base mutation, there can be numerous outcomes which are silent mutation. In such mutation, the effect is not observed in the polypeptide as a codon of an amino acid is changed to another codon of the same amino acid. Missense mutation. In such mutation, the codon of one amino acid is changed to a codon of another amino acid, leading to change in the polypeptide sequence. Nonsense mutation. In such mutations, there are insertion of stop codons in the coding sequence, leading to termination of a polypeptide synthesis. Frameshift mutation. In this type of mutation, the reading frame of the codon on an mRNA is modified, leading to defective polypeptide. Frameshift mutation can also lead to nonsense mutations. The severity of missense and nonsense mutation in physiology of an organism may differ with the kind of amino acid that is affected. As for example, substitution of a chemically synonymous amino acid in missense mutation may not have a profound effect on the polypeptide, whereas a chemically non-synonymous substitution can have a profound effect on the structure as well as the function of the polypeptide. In nonsense mutation that leads to premature termination of translation, there can be considerable effect in protein function unless the termination occurs very close to the 3' end of the open reading frame. This leads to production of a partially truncated polypeptide that may not have a distinguished effect on its function. As in nonsense mutations, addition of a single base or deletion affects the polypeptide sequence that is beyond the actual site of mutation. There is a change in the reading frame of codons present on the mRNA right from the point of mutation till the carboxyl terminal of the polypeptide, causing a change in the entire sequence of amino acids. Therefore, frameshift mutation leads to complete loss of function of a gene product. There are certain mutations which may not be in the coding region but may be present in the non-coding region. So let us discuss about mutations in the non-coding segment of DNA. Any part of the whole sequence of DNA can undergo mutation except at the non-mutable sites. Although we see a profound effect when the coding sequence are affected, mutations that occur in the regulatory and non-coding sequences can also have a distinct effect on the gene expression. The cis factors present on the DNA where trans factors binds are important regulatory region of gene function. Some of the important non-coding sequences are the ribosome binding sites of mRNA and self-ligating sites of Intron excision in eukaryotes. The consequence of mutation in parts of the gene other than the coding sequence are much harder to predict. In a more general statement, we can say that the functional consequence of such mutation in the non-coding region depends upon the location of the mutation, whether it is in a functionally relevant region or not. Mutations that disrupt these sites have the potential to change the expression pattern of a gene in terms of the amount of product expressed at a certain time or in response to certain environmental cues or in certain tissues. Looking into the factors that causes mutation, mutations can be classified as induced or spontaneous. Induced mutations are those that are induced purposefully with exposure to mutagens, whereas spontaneous mutations are those that arise spontaneously without intentional exposure to mutagens. Spontaneous mutations are the source of natural genetic variation observed in a population and have played vital role in evolution. In the next topic, we'll be discussing about induced mutation and mutagens. There are various forms of mutagens. The first one is base substitution. Some chemical compounds are similar to nitrogenous bases in the DNA and can be incorporated in substitution of the normal base. These type of compounds are called base analogs. Unlike the normal bases present in DNA, the base analogs have a different pattern of pairing and can induce mutation by allowing insertion of incorrect nucleotides during DNA replication in a process called the tautomeric shift. As for example, 5-bromouracil, which is an analog of thymine, promotes base pairing with guanine instead of adenine. In another form, called the base alteration, some mutagens are not incorporated into the DNA but instead alter a base, causing specific mispairing. Certain alkylating agents such as ethyl methanesulfonate, also known as EMS, and the widely used nitrosoguanidine, also called NG, are examples of such mutagens. Some other mutagens, also called the intercalating agents, are another important class of DNA modifiers. This group of compounds include proflavin, acridine orange, and a class of chemicals called ICR compounds. These agents are flat planar molecules that mimic base pairs and are able to slip themselves in intercalation between the stacked nitrogen bases of the core of the DNA double helix. In this intercalated position, an agent can cause single nucleotide pair insertion or deletion. Intercalating agents may also stack between bases in single-stranded DNA. In so doing, they may stabilize bases that are looped out during frameshift formation. In the base damage, a large number of mutagens damage one or more bases, so no specific base pairing is possible. The result is a replication block because DNA synthesis will not proceed past a base that cannot specify its complementary partner by hydrogen bonding. In bacterial cells, such replication blocks can be bypassed by inserting nonspecific bases. The process requires the activation of a special system, the SOS system. SOS induction is a last resort and used as an emergency response, allowing the cell to treat death for a certain level of mutagenesis. However, the repair mechanism is error-prone and can lead to mutation. In the next topic, we are discussing about spontaneous mutation. Spontaneous mutation arise from error in DNA replication, spontaneous lesions, as well as insertion of transposable elements. Spontaneous mutations are very rare, making it difficult to determine the underlying mechanisms. Some of the factors that can lead to spontaneous mutations are error in DNA replication. Mispairing in the course of replication is a source of spontaneous base substitution. Most mispairing mutations are transitions. This is likely to be because an adenine to cytosine or guanine to thymine mispair does not distort the DNA double helix as much as adenine to guanine or cytosine to thymine base pairs do. However, transversion also can occur throughout mispairing. Replication error arises due to low fidelity of DNA polymerase or an error in the proofreading activity. Naturally occurring damage to the DNA, called spontaneous lesions, also can generate mutations. Two of the most frequent spontaneous lesions are depurination and deamination. A mammalian cell spontaneously loses about 10,000 purines from the DNA during a 20-hour cell generation period at 37 degrees centigrade. If these lesions were to persist, they would result in significant genetic damage. However, most of it are corrected by DNA repair mechanism. The deamination of cytosine yields uracil. Unrepaired uracil residues will pair with adenine in the course of replication, resulting in the conversion of a GC pair into an AT pair. Deamination at certain cytosine position have been found to be one type of mutational hotspots. In conclusion, mutation is a process that is essential as well as deleterious. Some of the examples of diseases arising from point mutations are cystic fibrosis, sickle cell anemia, Tay-Sachs disease, phenylketonuria, and color blindness, among many others. Mutation is the source of variation in a population, leading to evolution, but can also have a negative effect if the mutation disturbs the normal functionality of the gene. Although the cells have its own repair mechanism that corrects changes in bases of DNA, error in the system can lead to undetected point mutations. Such point mutations in either the coding or non-coding segment of the gene has a profound effect on normal physiological function of a cell.